Hi, everyone. So I think we'll go ahead and get started so we don't take up too much of Polly's time. So it's my great pleasure to introduce Polly Fordyce, who's a very newly tenured associate professor of genetics and bioengineering at Stanford University. I first met Polly not too long ago when I was introduced by a joint collaborator of ours, Dan Hirschlag, and I was really impressed. Polly was telling me about all this great new technology in her lab, and I was telling her about my dream of opening a Mexican restaurant called Avocado's Number. And we uh, hit it off, and I think you can see from her training that she's done a lot of really hard quantitative biophysics, starting with her graduate work with Steve Block in the single molecule field, but then as a postdoc with Joe DeRisi and Steve Quake, really moved into high throughput measurements where you could still use the advances of microfluidics to make very quantitative and rigorous measurements of complex biochemical systems. And she's really pioneered that and has received many honors, including recently the Eli Lilly Award in enzyme chemistry from the American Chemical Society. So with that, I'll turn the floor over to Polly.
Okay, hello? Yes. Okay, so thank you so much for the opportunity to speak. And I just want to start a little bit by motivating the idea that physical forces at a most fundamental level are what determine the macromolecular interactions that determine life. So, we know that thanks to these physical forces, physical and chemical forces, a protein will either fold or not fold into a potential structure, and we can characterize this by an energy of folding. And then the folded or unfolded sequence will go on to do a bunch of different things in the cell, maybe interact with DNA for transcription factors or turn over particular substrates in the case of an enzyme. And whether or not these functions happen is a function of the conditions in which these reactions occur. So the concentration of the proteins has an impact, as well as the temperature and ionic strength, things like that. And then this entire sort of ensemble of interactions is what ultimately determines cellular outcomes like transcription or cellular signaling or all of metabolism. And so if there were any doubt that there's a really intimate connection between these fundamental physical forces and health at the level of cells or even organisms, you can just think about the human beta globin gene, where a single amino acid substitution from a charged residue to a hydrophobic residue means that these proteins assume the same structure, but only under conditions of low oxygen do you have it where the native protein is supposed to form these tetramers, whereas this mutant protein, these hydrophobic patches, drive the formation of fibrils, and then these fibrils lead to sickle cell diseases and all kinds of really terrible symptoms at the level of a human. So if we could kind of bridge this gap between fundamental physical forces and cellular outcomes, it'd be really great. In medicine, we could predict the functional effects of mutations or design novel therapeutics and diagnostics. In engineering, we could create enzymes that could do chemical reactions with less waste, or they could potentially clean up mining waste. But it's a really hard problem to get from all the way from these fundamental physical forces all the way up to cellular behaviors. And so you might think that it's totally an impossible problem, and I just want to make a quick analogy to another complex system where I think we've made really remarkable progress, which is thinking about predicting the weather. So somebody gave me a book a little while ago called The Weather Machine, which is all about the history of weather forecasting. And this guy, Lewis Fry Richardson, sort of realized that you could take somebody before him had realized that weather was basically just fluids in the sky. And Lewis Fry Richardson realized that you could break apart solving these couple differential equations into tiny little boxes, and that would allow you to make the problem easier such that you could begin to predict the weather. And he had kind of these really beautiful drawings where he envisioned there would be a central conductor who would be conducting computers, these are human computers, where each person would be assigned a particular calculation, and then all together they would be able to combine the outputs to predict the weather. And in his estimation, it would take 300,000 people 24 hours to be able to compute the weather 24 hours in the future. So it's going to take that many people just to predict things as they happened in real time. And he has kind of this beautiful quote where in 1922, he says, perhaps someday in the dim future, it'll be possible to advance the computations faster than the weather advances and at a cost less than the saving to mankind due to the information gained, but this is a dream. So that was 100 years ago. And then thanks to advances like the development of mainframe computers, we've now gotten to this place where even though people think that the weather forecast is not that good, it actually is very accurate most of the time. About 70% of the time, we do really well at being able to predict the weather. So what drove this incredible progress? It was advances in the actual hardware, the computational hardware, advances in the models of climate and the algorithms, but at a fundamental level, it was the fact that we had networks of weather stations all around the globe that were making measurements of the local weather using the language of physical and physical constants. So nobody was saying it's like twofold hotter here today than it was yesterday, or it rained 10% more than it did last month. Everybody was making measurements in absolute units, and that allowed us to combine measurements across space and time and conditions in order to develop these predictive models. So if we come back to this central challenge of trying to think about how do we get from underlying physical forces all the way to predicting, or at least accurately modeling cellular behavior, I think we've had a lot of really incredible advances in machine learning to try and predict the low energy folded structure from sequence. And I think that we're having all of these really beautiful tools for high throughput measurements of cell state. And what I think we really should focus on is making a lot of these measurements of protein function and stability using the language of kinetic and thermodynamic constants. And so that's sort of the main focus of my lab, and I'm just going to tell you two brief stories today of areas where we've tried to do this. My lab is primarily a microfluidics lab, and we have three main platforms that we use. One I won't talk about today is we have a way of making beads that have about 1,000 different embedded colors in them. So by attaching different things to the beads, we can keep track of 1,000 binding reactions throughout an experiment. And we also have these ways of making microfluidic emulsions that can be sorted using commercial fax machines for ultra-high throughput screening assays. But today, I really want to tell you two stories that use these valved reaction chambers that we can program with different protein variants. And so the first story is a very close collaboration with Dan Hirschlag's lab. That's how I met Aaron. And it was led by Craig Markin and Daniel Makhtari. And all of these people are fully joint between both of our labs. So it was a really close collaboration. And some of, the motivating question behind this project is the idea that an enzyme is more than an active site. If you just put active site residues in solution, you wouldn't get anything good. And so there's the whole rest of the protein. We know that some of these faraway residues play a really critical role in catalysis, but we don't really understand why or which ones. And so how can we probe this problem? If we start to think about enzyme phenotype as kind of a three-dimensional space, where along one axis we have every possible residue position in an enzyme, along another axis we have every amino acid that could be substituted or could exist at that position, and then along a z-axis we have some sort of phenotype. So perhaps the behavior in the presence of a particular substrate at a given concentration. So historically, with site-directed mutagenesis, we've chosen a few residues that we know are important, maybe the active site, and we've made mutations to those residues. And then for each of those mutants, we've collected a lot of information. So maybe we varied the concentration of a substrate and looked at the initial reaction rate as a function of substrate concentration. And from that, we can fit a Michaelis-Menten equation and get Kcat, Km. Maybe we can look at reaction rates in the presence of an inhibitor, so we could get the inhibition constants. We could get thermodynamic and kinetic constants that allow us to explain what we observed and predict what we might see under different conditions. More recently, I think there have been a lot of really impressive improvements in droplet technologies and sequencing that make it possible for us now to look at much larger sequence spaces. So now you can look at what happens with every single possible amino acid substitution at every position in an enzyme. But typically now, the information that you can get out is somewhat more limited. So perhaps you'll look at the fitness of an organism that contains a particular variant, or you could look at the amount of turnover at a given point in time. But I think what can be challenging is if you have a particular variant that's 10% down in activity, it could be 10% folded and 100% active, or it could be 100% folded and 10% active. And moreover, depending on the kinetic parameters of the enzyme that you're looking at, perhaps here's a wild-type enzyme and a mutant enzyme that have different Kcat and Km values. Under one substrate concentration, they have identical behavior. As you change that and look at another substrate concentration, you can see very different behavior. So what we were trying to see is, what if we don't make as many mutants? What if we substitute 2 different residues at every position in the enzyme? So we're going to put in a glycine, which gets rid of the side chain and increases the local backbone flexibility, and a valine, that's just like a regular-sized hydrophobic residue. We'll put each of these at both locations, but then we're going to go ahead and we're going to try and make all the measurements that we made before, where we'll vary the substrate concentration, get these Kcat, Km values, look at inhibition. So we have many fewer mutants, but we have a lot of measurements. So that technique we're calling HT-MEC, for high-throughput microfluidic enzyme kinetics. And this technique really relies on these 2-layer valved microfluidic devices that have underneath here 1,500 programmable reaction chambers, where the fluid flow within and between the chambers is controlled by 4,500 different on-chip valves. And so the way it works is we have to make a library of variants that we want to look at, with each variant in its own well of a multi-well plate, and we know who's who. And then we can use a robotic printer to print the DNA on a slide. So here I'm only showing you 3 spots. We have about 1,800. And the identity of the mutant is given by its position in the array. We can align a microfluidic device on top so that now each of these chambers is programmed with a different mutant. And now we can push in vitro transcription translation mix into all of the chambers. So we can make, and we take the device and we just sit it on a hot plate. So now we can make protein in every chamber in parallel. And I'm making this green here because we're going to make a protein that has a C-terminal EGFP tag. And then now we can pattern by iteratively actuating these valves. We can put an anti-GFP antibody via neutrabinin and a biotinylated BSA on the surface, such that we can recruit the EGFP tagged protein. And now we can trap that bound protein with these valves and wash. So we can wash out all of the in vitro transcription translation mixture. And this is just, I think, like the dirty secret of valved microfluidics is everyone always shows you the device, but they don't show you all of the stuff that you had to build to run the device. So I'm going to be really transparent about that. This is what the devices look like when they're hooked up with all this Tygon tubing. Sits on just a standard epifluorescence microscope, but then this little apparatus here is just a way to take pressurized air in from the wall, and then these are just banks of solenoid valves that either pressurize or don't pressurize these lines based on what the computer says in order to open and close valves. And so at the end of the day, do we make protein? Here's an image of part of the device after we've done our on-chip protein expression. We can zoom in, and you can see that some of these devices are labeled, some of these chambers are labeled. That's where we printed DNA. And we see these bright spots. Those are the EGFP tagged protein on the surface of the chambers where we made protein. These other chambers, we didn't print any DNA, we don't make protein. So that's a good sign. And I should also say we can calibrate these intensities so that now we know exactly how much enzyme we have. We know the effective concentration of the enzyme in each chamber. And then now we can take these surface-bound enzymes and we can introduce a fluorogenic substrate or coupled reagents for a coupled fluorescence assay. We can actuate these valves to start the reactions, and then we can sit and we can image the chambers over time, so we can watch as these different enzyme variants turn over their substrate and produce product. And so at the end of an experiment, for each chamber, we have a little plot that shows us what was the EGFP intensity for each assay that we did. We have a little plot that tells us what's the standard curve so that we can relate the intensity in that chamber to an amount of product produced. We have, for each substrate that we introduced, we can look at the amount of light produced over time and fit this initial rate. And then we can take those fitted initial rates and plot them as a function of substrate concentration, so now we can fit and get the Michaelis-Menten parameters. And so this is the report that we get for, and I should also say, we tend to go back and we iteratively, every 5 or 6 assays, we measure a reference substrate concentration, again, just to make sure that the enzyme is still behaving the same. So we can throw out a device. We stop using a device once it starts, the behavior starts looking funny. So this is what one chamber report looks like. And each time we run an experiment, we get about 1,500 of these, so we can run 10,000 reactions. And at the time when we had published this paper, we had run like 500 devices and 1 million chemical reactions. So before going on, I just want to pause and see if anybody has any questions about how the device works or the data analysis or anything before I show you any data that came off. Yeah, I'll go. Yeah, you can go first. Yeah.
That was super cool. I saw your pictures of all those valves and all those
All the tubes, yeah.
Is it really hard to make those? Like, do they all work the first time you try it? Or?
Yeah, so I would say it depends on how long you've been making them. So like a new student in the lab will run experiments for like 6 weeks without anything working typically. And then for the people that are better at it, for the people that have more experience at it, it's about 70% of the devices work, but 30% still fail. And it's still like you have to hook up, like you have to hook up all of those tubes by yourself. Like that, there's no like magic automation for that part. And the alignment is still by eye too. So there's a certain amount of craftsmanship, I would say, still in the experiment, which we would love to get rid of, but that's where it is now. Yeah.
Are these individual substrate reactions done in parallel, or are they sequential? Like you add substrate, wash, add substrate, wash?
That's exactly, yeah. So we add substrate, we'll monitor, we'll wash, we'll add substrate, monitor, and wash, and then we try and go back. Like, sometimes devices will last for a week, and you can get a huge amount of data. Other times, you go through all of the process of setting it up, and then you're just getting ready to go, and the device will kind of peel off the glass, and you have to start over. So when it works really well, but it doesn't always work.
Quick question.
Yeah.
Do you build in replicates in those wells?
Yeah. So the other things that we do is for these experiments, every 7th chamber is blank, and so we interpolate like we try and look and see what is the measured activity in these blank chambers, and that allows us to interpolate a local background. So we can say we're only going to consider measurements that are at least 5-fold higher than the interpolated local background as being real. And then we typically have like 4 to 5 replicates on a device. And all of these measurements happened in 3 stages, where we measured everything that was fast, and then there's some things that that they're going to be much slower. So we measured everything that was fast, then we got rid of that, and we measured everything that was slower. Now we have more replicates for the slow thing. And then there are still some things that were below the lower limit of detection, so we took those last stragglers, measured them on their own device, where they have like 50 replicates per mutant. Okay. So just to sort of hammer the point home, if we were going to try and make some of these measurements with traditional biochemistry, where we'd have to grow up culture and exchange buffer and then measure a different well, working pretty hard, it's very productive if you could do 5 enzymes in a week. But now we're able to do recombinantly express, purify, and assay 500 enzymes in about 48 hours, so that now it's really the library generation that's starting to become more rate limiting for us. Okay, so what kind of questions can we ask with this? And so a first question that we had was just how many residues alter function when you make a mutation in an enzyme? And so we're really fortunate in that the Hirschlag lab has decades of experience looking at these alkaline phosphatase superfamily members, which meant that we had a lot of background knowledge about what we were going to expect and a deep knowledge of the literature to interpret what we saw. So this is kind of an ideal enzyme system to start in some ways. It's a monomer, it's highly stable, it has a well-characterized reaction mechanism, and it also had fluorogenic substrates and existing crystal structures. So Daniel and Craig made a library where they put glycine or valine at every single position in path A, and then they measured just what's the impact on kcat over km. So on the x-axis, this is the log 2-fold ratio of the kcat over km for the mutant over the wild type. And you can see a very few mutations speed it up. This is consistent with just what we would expect for false positives. And so I don't think, it's a catalytically perfect enzyme. I don't think anything's actually speeding it up. Most mutations reduce kcat over km quite a bit. And here, this is the bootstrapped p-value with which we can say that it's statistically significantly different from the wild-type distribution. And if we look, we project these results back on the enzyme structure. Here's the active site here. You can see that, as we would expect, some of the residues closest to the active site have the biggest impact when you mutate them on kcat over km, but we see impacts that extend all the way out to the, to the peripheries. And overall, and you can see the same pattern. This is for the valine mutations. This is for the glycine mutations. And overall, about 2/3 of the residues, 2/3 of the mutations had a statistically significant impact on catalysis. So the next question is, why do these mutations impact catalysis? And what aspect of function do they alter? So one of the first things that you might think is maybe all of these mutations are just unfolding the enzyme, right? So we always kind of think about this Michaelis-Menten scheme, but really enzymes exist typically in an equilibrium between the folded and the unfolded state, and maybe we just destabilize the protein. This is a super stable secreted phosphatase, so it seemed unlikely that a single mutation would do that. But to test, we can just remeasure the rate, and we can compare the rate relative to the wild-type protein at 0 molar urea and the rate relative to the wild-type protein at 2 molar urea. And if it was significantly destabilized, now we should expect that in the presence of 2 molar urea, it should be essentially dead, but it's not. So that means that we have not, under equilibrium conditions, we haven't destabilized the folded protein. There were some pieces of evidence that I'm not going to talk about now that there was some sort of defect in folding. So this led us to ask, is there some reason that we could be changing the folding pathway of this protein? So to do this, we have to remember not only that the enzyme exists in an equilibrium between the folded and unfolded forms, but also that you have to get to the folded form, right? You have to start unfolded, and you somehow have to fold properly. And so one way that we could test if mutations are altering this folding pathway is we could vary the zinc or the temperature. The zinc concentration, it's a bimetallozinc enzyme. I should have said that earlier. And we could vary the temperature, and we could see whether that alters. If we vary the expression conditions, and then we assay under identical assay conditions, we should be able to see if there's something going on in the folding pathway. And so that's what we did. And now you can see there are many mutants that when we express at lower temperature, they're almost wild-type-like, whereas they're really down when we express at high temperature. And same with the zinc. As we add more zinc, we can rescue a lot. And again, we're assaying under identical conditions. So we can see what parts of the enzyme are responsible for these properties, and we can see there's some structural zincs up here. So you see strong zinc effects in this part of the enzyme. The overall enzyme core is sensitive to differences in temperature. And we can ask, is this something that we're just seeing on our microfluidic device because we're using cell-free extracts and it's a microfluidic device, or is this actually a property of the protein? And so we took the wild-type protein, Fanny did this, in several of these mutants. And if we express at 23 degrees C in E. coli, you can see that we just see this single predominant band on a native gel. That's the properly folded protein. If we raise the temperature, now you can see that the wild type is still primarily in the native fold, in their native form, but these other mutants now have the appearance of this misfolded band. And so a question that we could ask is, for this misfolded form, is it catalytically active or not? And to test that, Fanny measured the activity of these enzymes before and after adding thermolysin. So when she adds thermolysin, she sees a disappearance of the misfolded band. So here's the activity prior to adding thermolysin, and here's the activity after adding thermolysin. So all of this together suggests that some of these mutations promote the formation of a long-lived catalytically inactive misfolded state, and we can quantify via some measurements I'm not going to talk about now how much of that protein is misfolded. So now we can drill down and ask, okay, now that we can get rid of all of the folding effects, can we ask how mutations are impacting catalysis? And so Dan has previously used these two different substrates. PAF-A is a preferential phosphate monoesterase. So for the monoester, these two residues, R164 and K162, make contacts to this phosphoryl oxygen. So if you mutate these residues, you should see a really big effect. For the diester substrate, these two residues, this oxygen is methylated, and so these residues don't make any contacts. So now if you mutate these residues, they shouldn't really have an effect on diester hydrolysis, but they should have a big effect on monoester hydrolysis. And here's just to orient you, these residues lie at the end of this specificity helix here. So when we repeat the measurements on chip, we see what we expect. So these are K162 and R164 mutants. On the y-axis, this is diester hydrolysis. They're actually a little faster than wild type in diester hydrolysis, but they're way down in monoester hydrolysis. Now we can look, what about all the other residues in the protein? So now you can see most mutations have equivalent effects on both monoester and diester hydrolysis, but a lot of these mutations look kind of like K162 and R164. So it implies that they're functionally coupled in some way to K162 and R164. And when we look at where these residues lie, they're second shell residues that are contacting K162 and R164. So maybe this points to a way where you could make mutations that would alter specificity potentially without having to mess with the active site. If we go all the way out to the surface, if you go all the way out to the surface, there are residues that lie on the surface that have 10 to 100-fold effects on the specificity, which suggests that maybe this is a way that you could be mapping potential allosteric interfaces that you might want to target for allosteric inhibition. And I just want to say, we can look at different aspects of function. For the sake of time, I'm not going to talk today about measurements looking at ground state destabilization. But we begin to see these large swaths of residues that are physically contiguous, extend far from the active site, and affect a given property of catalysis. And different properties seem to map to different parts of the enzyme. So, from using this device, we were able to, start with an overall map of KCAT over KM effects, deconvolve them into folding versus, effects on the chemistry step, and then further map out how different mutations were altering substrate specificity or specificity for the ground and transition state analogs, as well as how temperature and zinc impact the folding pathway. So one of the next questions that we wanted to ask was, if we try and probe further, we've looked at substrate specificity and we've looked at ground state destabilization. What else? We know that an enzyme has to preferentially stabilize the transition state in order to drive catalysis. So what's responsible for that preferential transition state stabilization? And so Craig and Daniel teamed up again. And this is now on bioRxiv, I should say, to try and look for the same mutant library. Can we learn anything using transition state analogs? And I think there's kind of a beautiful quote from a paper earlier last year. If we're to overcome our reliance on high-throughput experimentation, which I hope we ultimately can, right? That's what we all want. We must consider the factors that make enzyme design so challenging. Efficient protein catalyst requires an extremely high degree of precision to achieve effective discrimination of the transition state from the ground state, and even angstrom-level inaccuracies in side chain positioning can have a catastrophic impact on catalysis. So what residues are responsible for this angstrom-level side chain positioning? So transition state analogs are a way that we can probe this. And for PAF-A, PAF-A is an interesting model system for this because the active site uses the exact same residues to contact the substrate in the ground state and the transition state. And this transition state analog is thought to pretty well mimic this actual transition state of PAF-A. So if we just look at the active site, as as has been seen for many enzymes for a long time and is predicted by a lot of theory, if you look at the impact of mutations on transition state analog binding and catalysis, for the active site, you get about the same amount of an effect, right? If you disrupt catalysis, you also disrupt transition state analog binding. And this is sort of consistent with a model where the active site residue is either there or it's not. If we move out and we look at the residues that contact the active site residues, now we start to see two types of behavior. So for some of these residues like this, the impacts are still equivalent, where you could kind of picture this as there's a mutant that's required to position an active state residue, and when it's gone, it's changing the fraction of time that the active site residue is competent for catalysis or not. We also, though, see the emergence of this new population of mutants that dramatically impact catalysis but have no impact on transition state analog binding. And as we move out in the enzyme from the active state to the surface, we see more and more of these residues where there are lots of residues that you can make a mutation and it ablates catalysis, but transition state analog binding is just fine. And you can sort of see if you project these two different populations on the enzyme close to the active site, all of these residues have equal impacts on catalysis and transition state analog binding, but a lot of residues all the way to the outside impact catalysis alone. And just to think about this angstrom level positioning, I think it's actually more complicated than that. It's actually sub-angstrom level positioning, because if you repeat these measurements with tungstate or vanadate, which differ by like 1/10 of an angstrom, they're like tiny, tiny molecular differences, you can see that there are mutations that, same with the vanadate case, they can ablate catalysis while leaving tungstate binding untouched, but they can even ablate catalysis and increase affinity for tungstate. So together, when we look at this map, I think that what it means is that some of these faraway residues are really important for discriminating substrates with less than 1/10 of an angstrom precision, right? So I think that means when we think about enzyme design, we're really having to think about the role of the entire protein, and that's why it's such a hard problem and a much harder problem compared with trying to engineer a binding interface. And just to try and reconcile this, we have to go from these kind of conformational coordinate diagrams where they're relatively simple to thinking about the idea that you can have an ensemble of a bunch of different microstates, and then different microstates have different capacities for catalysis and binding. Okay. So, in the future, we're really interested in trying to use this platform to characterize enzyme variants, to understand their effects for precision medicine, try and see whether we can systematically discover interfaces with allosteric potential, work on iterative testing of designed enzyme libraries, as well as to try and work with our computational colleagues doing MD-based simulations to try and provide ground truth data that could assess and ultimately improve those models. But in my remaining time, I just want to tell you one other quick story that's not about enzymes, and it's instead about transcription factors. And this is a project that was led by a really talented technician who joined my lab right out of undergrad and stayed for a few years, and he's now a graduate student in the program at Berkeley. And it's been a really nice collaboration with Anshul Kandaji's lab and Julia Zeitlinger and Raluca and Will. And it's on bioRxiv for forever because I can't get it published. I think it's really cool, and so I'm hoping eventually somebody will like it. Okay. So it starts with the observation that the human genome is filled with repetitive elements. So when you consider protein coding genes are about 1.5% of the genome, simple sequence repeats are twice that. And these simple sequence repeats are short tandem repeats with repeated units less than 6 base pairs. So they look kind of like that. We know that they're enriched in enhancers, and we know that they alter transcription. And there are a lot of papers that have tied the presence or absence of these short tandem repeats in enhancers to disease and to changes in gene expression. But what we don't know is why these short tandem repeats exert their effects. And people have hypothesized that maybe it has to do with nucleosome positioning. But we were really curious, what about at the most fundamental level do short tandem repeats alter transcription factor binding? And so we took 2 model systems we use a lot in the lab. These are 2 basic helix-loop helix transcription factors, FO4 from yeast, which responds to phosphate starvation, MAX from humans, which is a kind of a signaling node. These transcription factors bind DNA in a really similar conformation, and they both really like this canonical E-box consensus motif, a CAC-GTG motif. And so one of the first questions that Connor wanted to ask was, does the presence or absence of short tandem repeats impact transcription factor binding by FO4 and MAX? And so he used these same devices in a slightly different way, where now he's going to print fluorescently labeled DNA at different concentrations and use that to try and come up with concentration-dependent binding curves to estimate affinities. And so he took a library of DNA that has a CAC-GTG in the middle and the same 2 nucleotides on either side, and then he surrounded that consensus site with either random sequence or different types of short tandem repeats, homopolymers, dinucleotides, tetranucleotides. And then he measured concentration-dependent binding curves for each of these sequences. And this is just sort of an example of what the data look like. So this is the CAC-GTG with a random sequence and with a repetitive sequence. And this is the amount of DNA bound per amount of transcription factor on the surface. And you can see we see about a 2-fold benefit for having this particular repeated sequence surrounding the consensus site. If we do this for all of these possible repeats, now we can see this is FO4 is in orange, MAX is in blue. And you can see that for some of the most favored repeats, you have about a 1.7 kcal per mole energetic difference between having random and repetitive sequence. So that's about a 25-fold change in binding. And there are also some of these repetitive sequences that penalize binding. They disfavor binding. And even though FO4 and MAX have the same consensus site recognition, they have different preferences for repeats. Okay, and this is if you take the position weight matrix model, the most popular model for predicting binding energy, and you scan it across the sequence and you ask what you should expect, that's these gray bars here. So these sites don't have, these repetitive sequences are not predicted to bind because of the presence of half sites or anything like that. Okay, so why are repetitive sequences altering transcription factor binding? So we were thinking about it in terms of 2 different models that we're going to explain with food analogies. So the first is there's something about the repetitive sequences that change the conformation of the consensus site to make it more appealing. So if this was like a breakfast muffin in the presence of a random sequence, and then we put repeats on the other side, the breakfast muffin has turned into a delicious cupcake, and it's more preferred for the transcription factor. So in this case, the repeats shouldn't have any impact in the absence of a core consensus, and there should be no change in the stoichiometry, the number of transcription factors bound. For a second model, the repeats themselves are bound by the transcription factors. So in that case, thinking about our muffin model, now the repeats are like a series of mini muffins that are flanking the main muffin. And then we would predict that the repeats should have an effect in the presence or absence of the core, and that the stoichiometry should change. So Connor did the experiment. He took away the motif, and he just measured concentration-dependent binding for these 2 cases. And the data are very noisy now, but we consistently see that the presence of a favored repeat leads to an increase in binding relative to the random sequence. And then he went and he ran some gel shift assays where this is fluorescently labeled duplex DNA. And then up here, you can see super shifted DNA that has been bound by a MAX transcription factor. And you see when we flank the motif with a random sequence, we see this amount of super shift. When we repeat the experiment, the concentrations are all the same, but we add repetitive sequences. Now we see an additional shift. So there's the formation of a higher order molecular weight complex that has further retarded the DNA in the gel. Okay, so both of those things appear to support our mini muffin model that the transcription factors are binding the repetitive sequences. Why do they do that? And I think that we can kind of remember that there's both, when we think about our observed Gibbs free energy, it has an enthalpic term and it also has an entropic term. And as Boltzmann engraved on his tombstone, that entropic term depends on the number of available possible microstates. And so when we think about the number of microstates that are available to these transcription factors, if you happen to prefer a homopolymer, that's pretty hard to, like, nobody really prefers a homopolymer, so we don't really tend to see that many preferred homopolymeric sequences. But these dinucleotide repeats give you a large number of potential preferred sites that you could sit down on, far more than if you had a sequence that's not repetitive at all. Okay, so the next question is, what part of the transcription factor is important for recognizing those repeats? And so now Connor went ahead, and he reused these microfluidic devices, kind of like we did for the enzyme experiment. So now he's going to mutate the transcription factor and ask how those transcription factor mutations impacted recognition of repeated sequences. And so he created 180 single amino acid substitutions within this FO4 DNA binding domain. And then for each, he measured concentration-dependent binding curves for the core surrounded by random or repetitive sequences. And this is just a sample of 3 of the binding curves that you might see for wild type. Here are 2 mutations that reduce the affinity. Okay, so now if we look at, this is a complicated plot in that the y-axis is showing you the impact of a transcription factor mutation relative to the wild type when binding the repeats. And the x-axis is the impact of a transcription factor mutation relative to the wild type when binding the random sequence. All of the transcription factor mutations have the same effect on both sequences except for E259D, which is kind of a clear outlier here. If we go back and we look at where E259D is, you can see that it directly contacts the DNA. And when we compare measurements of KDs for the wild type protein interacting with 6 different single nucleotide substitutions and the E259D, you can see that the wild type has this strong preference for the consensus, and it really disfavors these other sequences. Whereas E259D, this is above background binding, and it doesn't really care. It's pretty promiscuous for any of these sequences. So we think you can kind of think about this like a model where, in a reaction coordinate diagram, E259D has kind of a flatter energy landscape, and now these repeats are closer to something that it might prefer. Okay. What about kinetics? Do the repeats matter for the kinetics of transcription factor binding at all? To answer this, Connor took these same libraries, and now he used the same devices to monitor the loss of DNA after we've come to equilibrium. And so we can get curves that kind of look like this for each of the different mutants interacting with these different, like the wild type proteins interacting with all of these different DNA sequences. And when we look across the different DNA sequences, for both of our proteins, this is the apparent macroscopic K-off. I don't know where it was bound. It could have been on the flanks. It could have been on the consensus. But the apparent macroscopic K-off is identical across all these sequences for both MAX and FO4. But we can see that particularly for MAX, there's a really strong impact on the K-on. So we see a much faster association in the presence of some of these favorable motifs. And that change in the apparent association is what's driving the difference in KD. We can develop a 4-state kinetic model in which the transcription factor is either really far away from the DNA, it's close to the DNA and kind of, but non-specifically bound, or it's bound to the motif and it's bound to the flank. Here's our observable K-on and K-off. And then we're able to fit, using some of our measurements, we can fit microscopic rate constants to that 4-state model and try and simulate what do we expect in a cell where there are multiple transcription factors. Should this impact transcriptional regulation? And what we see is if we look at the dwell times of the transcription factors between these 3 different repeated sequences, it's not different. Like an individual transcription factor is on or off for the same amount of time, consistent with the fact that our observable macroscopic K-off didn't change. But from the perspective of the DNA, having a bunch of transcription factors bound on the repeats means you have kind of a reservoir of transcription factors that's available to get to your consensus site. And so you can kind of see here, this is the mean occupancy. At the motif, it stays constant. at the flanks, and at total, you can see you start to have more transcription factors on your DNA as the repeats become more preferred. And that means that your mean free passage time, the amount of time that it takes for you to get a transcription factor on your consensus, goes down. So this could be, the presence of repeats could be a way for cells to make it possible to turn on the expression of a gene faster. How do we know if this matters in a cell or not? So I think this experiment is hard to do, and so we turned to this, but I'd be happy to talk to other people about this later. We turned to a bunch of existing ChIP-seq data that look at where transcription factors are in cells that were measured by a whole bunch of different labs. And then we teamed up with Anshul Kundaji, who has developed some deep learning algorithms that can learn from those observed in-cell occupancy profiles to predict transcription factor binding and nucleotide resolution on an arbitrary sequence. So these algorithms have only seen the cell-based data. They haven't seen any of our data. And then we just asked, if you take this trained algorithm and you ask it, what does it think should happen when we give you some of these sequences that we've measured? What are your predictions, algorithm? And so, first we asked it kind of an easy case. If you have sequences where you have a motif, a mutant motif, or no motif, do you get that the motif matters? So the x-axis is the measured binding affinities where stronger interactions are lower delta-delta Gs. And here's the predicted occupancy, so higher means more occupancy. And the algorithm realizes that the motif matters. Now what about if we ask, what about the motif if we surround it by different types of repeats? You can see that the algorithm has understood that some of these repeats promote stronger binding, even though it's never seen any of our in vitro data. And when we try and interpret what was important for these predictions, a lot of the time the algorithm is sort of revealing some of these repetitive elements that are upstream or downstream of the known consensus. So it seems like, I would argue, if the algorithm learned how to predict things really well in vivo, and it knows that this is important for binding in vivo. And we can also, we can do the same thing where we can ask, does it matter how perfect the repeats are, or is it just the nucleotide composition? So we can increasingly scramble these sequences. And both our measurements here, we can see that that scrambling does have an impact. It's not just the nucleotide composition. And then the algorithm predicts the exact same thing that we see in our measurements. So we've been sort of excited about this idea of, can we use machine learning as a way to do ultra-high throughput biophysics in silico so that we can identify some of the areas that are most important for us to measure in vitro and try and understand the underlying physical mechanism. Maybe this is just for FO4 and MAX and it doesn't matter for any other transcription factors. We're super fortunate that labs like Tim Hughes and Martha Bulik and Raluca Gordon have spent a lot of time collecting really beautiful protein microarray data where they have many different sequences on a slide, and then they quantify how much of a transcription factor binds to those sequences. So we can take those data, we can consider there are 65,000 possible 8-mers, and we're just trying to try and calculate for every probe that has a given 8-mer, what's the median intensity of all those probes. So we get a distribution of intensities where this is the number of 8-mers with a given intensity. Now we can calculate a z-score for a given 8-mer. This is what these z-score distributions look like. So as you would expect, z-scores that are really high, it's bound way above background, those are all for FO4 and MAX from the protein binding microarray data. Those all have some CAC GTG in it. Those are all consensus sequences. But if we zoom in on this background binding, you can see that many of these repeats, and these are the favored repeats in our experiments, are bound above the background levels. So this very weak preference, when combined with many possible ways in which you could bind, can lead to an appreciable increase in affinity. And if we go back to the standard models, like the position-specific affinity matrix versus the measured delta-delta Gs, you can see that there's no real strong correlation between the standard model predictions and the measurements. But if we use these kind of median intensity values, these intensity z-scores, as a way to calculate a partition function, now we can see that our partition function prediction really recapitulates the measurements. So this is just an additive model that takes into account very weak affinity preferences for other sequences that aren't the consensus. If we look over all of the data for all transcription factors, there's like 1,000 transcription factors. This is all of these transcription factors across the columns and coming down on the rows. These are different 8-mers. And here's the calculated z-score, the degree to which that particular repeat is preferred. Many transcription factors have a strong preference for a particular repeat. Sometimes it's trivial. Sometimes it's just like for a high mobility group transcription factor, they prefer consensus sequences that look like repeats. So it's not at all surprising that they're sensitive to these repetitive elements. And we can kind of quantify that by saying the z-score as a function of the Levenshtein distance, which is just the number of mutations away from the consensus site. In this case, you see a trend like this. It means they like repeated things, and there's nothing special or interesting about this. But for a lot of transcription factors like FO4, you see these more complex patterns where they have a strong sensitivity that isn't really predicted by any kind of closeness to the known consensus site. So why do I think that this happens? If you think about, here's an example of 3 transcription factors from mice that have the same consensus, but when you look at their z-score sensitivities, it's very different. So if you look at their predicted binding to different sequences, it's very different. Is this a way that evolution can allow closely related paralogs to regulate distinct targets, distinct transcriptional networks? So I just want to conclude by saying I hope I've convinced you that transcription factors can directly bind short tandem repeats that might or might not look like the consensus. The DNA binding domain is what does the binding, and that they can reduce the mean free passage time to the consensus, and that this is a widespread phenomena. I want to thank all of you for your attention and for the invitation to come speak, and everybody in my lab. And I'll take any questions if you have them.
Thanks, Polly. Are there any questions? So maybe I can start off. So the transcription factor data, I was kind of wondering if the proteins are kind of binding at the repeats and then sliding to the consensus. And I'm wondering if you've thought about sliding or tested like salt dependence or anything.
I would love, so our model, that's like a really obvious transition that's missing in our model. And it's just missing because we didn't have a way to get rid of sliding and see how things were impacted. So a really obvious thing to do would be like to move it further away, move the repeats further and further away, as well as put like a dead Cas9 or something that would block sliding and mean that you had to detach. I would love to do that, and I think it's really interesting, but we haven't done it yet. It's a great question.
Anyone else? Bob.
I think maybe similar vein, just trying to wrap my mind around what's going on in these repeat sequences. If I followed, you think for the high affinity cases with the repeat sequences, you may be getting multiple proteins bound. You have multiple signals. So I'm kind of curious in where you're looking at the off rate, if you're saturated the binding and if there's more than one protein bound.
Why don't we see a chaos
Do you see
effect?
biphasic or single phasic?
So in our experiments, there's only, like because we're on a surface, we only see, in our kinetic measurements, we only see the interactions between one transcription factor and the whole DNA. It looks like a single exponential, that bulk behavior, but we don't see, I think when you think about it in an actual mammalian cell, I think it's more important in mammalian cells than in bacterial cells. And in that actual situation, you have a bunch of transcription factors that are likely contacting co-activators via activation domains where they might be linked into larger complexes. And then I think that could lead to having much longer dwell times in vivo than what you would see in vitro, because you have kind of this multi-domain interactions. For us, we don't see an off-rate effect here because we're only looking at a single transcription factor at a time, if that makes sense.
So you think you're just seeing the single protein bound to the cognate site?
Well, or it could be, I don't know where it's bound. I'm seeing the bulk population
Yeah.
that could be either bound to a repetitive sequence or it could be bound to the consensus sequence.
But if they have a single off rate, then that would have to say the off rate's independent of sequence, right?
Yeah, assume we put in dark competitors, there should be no rebinding. But I guess I just talked to Zev Bryan about this last week, and he was like, have you thought about the fact that your dark competitor could also be driving like facilitated displacement? So you could not be seeing what you think you're seeing with the dissociation, which I hadn't thought about before.
Yeah, so that, I mean, maybe that gets it. So what I was trying to understand is if you see an effect on rate, and it's because you can contact the adjacent sequences and slide or whatever, why you don't see the reverse of that in the off? That is, it would microscopically release, why wouldn't it sample the sequences and have the same effect?
So you have to, I think it's because it's not, you can no longer think about it like a two-state model anymore. And so you end up having, like this behavior is predicted when you try and analytically solve the equations from that four-state kinetic model to predict what you should see for K on and K off. It is predicted by this, but it's no longer, I totally agree, if it's two-state model, it should be, if it speeds up K on, it should speed up K off. But when you add the additional state, it no longer does that anymore. Yeah.
Okay, thanks.
But the model is I think sliding is exactly what we haven't seen. Yeah.
Any other questions? So, Jim.
So excuse me, in your experiments, you always have the same sequence, flanking sequence and the repeats. What happens if you have different sequences on one side or the other? Are they additive?
That's a good question. We have some sequences where, I don't know if we have every case. Some sequences, some repeats have different sequences on one side versus the other, but I don't know if we have this, I don't know if we can compare it with the same sequence on both sides to see if it's additive or not, but that's a very good question. I haven't thought of that before. It's a very good question.
Any other questions? All right, Phil.
You saw a number of mutations, going back to the enzyme, that seem to enhance the transition state analog binding, or keep it the same, but decrease catalysis. Could that be the result of changes in the conformational dynamics and being able to cycle through the product?
Yeah, I guess one question is, do you have to invoke dynamics or not? And you know, this is like a very controversial area, and I guess, you know, I'm like new to it. It seems like you could just say, it seems like you can explain it by equilibrium, where there's just a slightly different active site configuration. Particularly the tongue state is like a little bit bigger. So now the fact you've made something that's a little bit better at binding tongue state, but a little worse at catalysis, I don't know that it requires dynamics to say that. I think it could just be like a slightly different shape of the conformational ensemble, and it doesn't have to deal with like the dynamics of changing between one state and another.
Dynamics could explain it, right?
The dynamics have to be happening. Things are moving.
Yeah.
Huge amount of thermal energy, right?
Yeah. I mean, I guess I'm agnostic. We don't have any NMR that would say it. I mean, Dan feels very strongly, actually, this is an area where Dan has a very strong feeling that you don't need to invoke dynamics, but I don't know that, like, he would make that argument more than me, yeah.
All right, let's thank Polly for the great seminar.